Hello everyone and welcome to Storm's Garden. Today we're going to be going over Chapter 4, Archaea, in our Teeming with Microbes book. Thank you for coming on hanging out. Let's get you set up and we'll get this show on the road. Alright. Archaea Bacteria. Archaea. I just googled Archaea. It says it right here, actually. Just a few years ago, no one would have thought to include Archaea in a book on the soil food web. These microorganisms, at first considered a weird and unusual subset of bacteria, were known to live only in extreme environments, such as around geysers or in hot water ocean vents. Since these spots don't exactly feature in our agricultural or horticultural areas, Archaea were not considered soil food web members. Then, at the turn of this century, directly as a result of advances in genetic identification of microbes, Archaea were found in soil. Moreover, it appears that the role they play in nitrogen fixing, taking atmospheric nitrogen unavailable to plants and converting it into plant usable form, is a crucial one. Now they have our attention. Backstory. Archaea were first identified in the early 1970s by Carl Woese, W-O-E-S-E, Carl, W-O-E-S-E, and his research associate George Fox, who were studying bacteria in the University of Illinois. Their interesting conclusion: bacteria could be divided into two distinct groups, normal bacteria and a new second group of extremophiles. They not only survive but thrive in high temperature environments. The second group produced methane, like the bacteria in the large intestines of humans. But most important, these new bacteria had a distinct genetic makeup. Excuse me for my sniffly nose. Not only had a distinct genetic makeup, it was shortly found that not only was the genetic makeup of all Archaea bacteria, as Woese first called them, different from other bacteria, it also differed greatly from the genetics of eukaryotes, organisms proposing cellular membranes, distinct nuclei, and cytoskeletons. Nice. I'm gonna blow my nose real quick. Nuclei and cytoskeletons, be they protists, fungi, plants, or animals. So eukaryotes have all of these things in their... There we go. So we have eukaryotes. Have basically all living things. We are eukaryotes, I think. Maybe. Bacteria was dropped from the name altogether. Uncovering a new form of life so late in the history of biology was a stunning development. It led Woese and Draw Wars to draw a new tree of life with three main domains or kingdoms: bacteria, archaea, and eukaryotes. Many believe that the kingdom Archaea is the oldest of the three branches, since the conditions in which many Archaea live are not thought to be representative of the extreme conditions that exist during the establishment of life on Earth. Bacterial lookalikes. On the matter of distinguishing between Archaea and bacteria, the word impossible comes to mind. It doesn't help that they are the same size, that is to say, really small. As with bacteria, as many as 500,000 archaea can fit inside the end of a period at the end of this sentence. Most archaea and bacteria look alike, so much so that using the eye, even at high magnification, doesn't reveal any differences. Like bacteria, archaea usually travel by whipping out one of their phalangula, and when they are not traveling, archaea too can form large colonies, though some archaea live a more solitary life, as do some bacteria. Archaea grow and multiply just like bacteria. They divide. One study suggests archaean cells divide every 20 minutes in ideal conditions. You don't have to do the math to know that in a short time the world would be a very different place if ideal conditions existed. Archaea often exhibit the same three basic shapes as bacteria, though those in the salt-loving genus Haloquadrum, let's see, Haloquadritum, are square, a shape not seen in bacteria. There are also needle-like filament archaea that look similar to actinomites. Some form perfectly rectangular rods, and some archaeans use the alternative teriki, plural form for them, are irregularly lobe-shaped, triangular, and even shaped like teacups. What makes archaea unique? With so much similarity, why did Wursch find reason not only to distinguish archaea from bacteria, but to declare them a whole new kingdom, a separate branch on the tree of life? A separate branch on the tree of life. To begin with, although both bacteria and archaea have cell walls that hold in and protect material inside, the cellular membranes of archaea contain either lipids, while bacteria cells do not. Archaea walls contain amino acids and sugars that differ from those found in the cell wall of bacteria, and unlike eukaryotes, archaea cells walls never contain chitin, as do those of fungi, nor do they contain cellulose, as do those of plant wall, as do the cell walls of plants. This is complicated stuff. With keratins, one of us is back to college. One of us back to college organic chemistry. The reason he is not a physician. Obviously, it takes computers and analysis well beyond the capabilities of the gardener to discern these differences. Trust us as we do Carl Warsch and his colleagues on the cellular level. These distinctions are so important, they are sufficient not only to split these weird, sometimes oddly shaped organisms into their own group, but to put archaeans into their own kingdom. The second characteristic that separates archaea from bacteria has to do with genetics. Both archaea and bacteria are prokaryotes. Their genetic material is not enclosed in a nucleus. However, the genes of archaea, but not bacteria, are more closely related to the genus of eukaryotes. those organisms whose DNA is enclosed within a nucleus. Specifically, in both archaea and eukaryotes, similar enzymes are involved in the synthesis of ribonucleic acid, or RNA, which itself is a key to synthesis of proteins. Another set of characteristics set archaea apart from both bacteria and eukaryotes. Archaea use many different substances for energy: sugar, ammonia, hydrogen, and many metal ions. Some can even use sunlight as a source of energy. Others use carbon dioxide. And while some bacteria and eukaryotes can reproduce by spores, archaea don't have this ability. Oh boy. I'm going to have to go get me some antihistamines. Oh goodness, I lost my page. Finally, if you really want to get down to detail in differences between archaea and bacteria, while their mode of transport is the same, flailing flagella, the chemical composition of the flagella and the mechanism that makes them operate is different. Talk about splitting hairs. Types of archaea. Determining the difference between a bacterium and archaeon then requires work on a genetic level. We think of it as just like reading barcodes. Each organism has a unique code. Using DNA and RNA sequencing, scientists can read the genetic makeup of a given sample, the barcode, just like the checkout clerk at the grocery store. These techniques work on DNA found in liquids, solids, and gases. Thus, all sorts of environments can be assessed, including soil. So far, approximately 250 different kinds of archaea have been found. More will be found now that scientists know what they are looking for and have a proper tool to do the job. Archaea play a big role in the carbon cycle, and one way to group them is to look at how they get their energy and nutrients. Some archaea are photoautotropic, obtaining energy from light and using CO2 as their source of carbon. Others are chemoautotropic, using inorganic chemicals as their energy source, while still employing CO2 as a source of carbon. Yet, others are photoheterotropic, which means they use light for energy, but not organic chemicals to obtain carbon. Finally, some archaea are chemoheterotropic. Whew. Using organic chemicals for energy sources as well as for carbon. Archaea are found in hot springs and salt lakes, living in rock buried deep in the earth, under miles of ice, and in hot deserts, environments inhospitable to most other living organisms. These extremophiles are able to survive in such environments because they have developed specialized enzymes that work at high or low temperatures, and because their proteins can fold tightly to avoid destruction by salt, heat, or cold. Special characteristics of their cell membrane also help make them resistant to heat. Archaeans that live in these habitats don't need oxygen. They are all anaerobic. Some archaea, known as acidophiles, have made cellular and metabolic adjustments so that they can live in extremely acidic environments with a pH as low as 1. Wow. Let's take a look at these quick. Ooh. And then we have another picture right here. Electron microscope. Heat-resistant enzyme complex from the extremophile archaeon. Function at 275 Fahrenheit. Wow. Amazing. They constantly pump hydrogen ions out of their cells, maintaining a much higher and only slightly acidic internal pH of 6.5. Another group of archaea are classified as halophiles, or salt lovers. Thermophilic archaea live at high temperatures, and the cold-loving or psychrophilic archaea live in frozen areas. Many of these extremophiles are also methane agones. Methanogons. Methanogons. Methanogens. Their metabolic activities produce methane, but may be a surprise to learn they smell that smelly swamp gas and cattle flatulence thereover thought to be produced by bacteria are actually the byproduct of archaea going about their daily activities. Methanogenic archaea are found throughout the oceans. In fact, so many of these organisms thrive in ocean plankton. It is now estimated that the combination of archaea in the ocean and those in soils make these microorganisms the most abundant life on Earth. Harnessing the properties that enable archaea to survive harsh and inhospitable conditions could have great economic consequences, but techniques for culturing archaea are still in their infancy. We are certain this and other difficulties will be overcome, and archaea will become important biotech tools. Archaea and the nitrogen cycle. To bring all this back to the subject at hand, it turns out that the archaea also participate in the nitrogen cycle depicted on page 48. This discovery was made in 2 steps. First, scientists isolated from ocean-dwelling archaea and specific gene that produces the enzymes needed to oxidize ammonia. When they looked for the same gene in soil samples, they discovered it was a whopping 3,000 times more prevalent than the counterpart gene in bacteria. They also looked at the amount of lipids associated with the organisms in the soil and confirmed the presence. And more astounding, the dominance of archaea in the nitrogen fixing process in soil. Even more interesting, the greater number of archaea continued at deeper soil depths, whereas the bacteria count petered out with depth. Samples taken around the world showed the same results, leading many to conclude that archaea, specifically the Centricaeota, Crenarchaeota, Crenarchaeota, are the most abundant and ammonia oxidizers in soil. This discovery is an important one, if for no other reason than nitrogen gases include nitric and nitrous gases. These are greenhouse gases, and it makes sense to figure out what proportion of them may be traced to the activity of archaea. There is still much to learn about the size and scope of the role archaea play in the nitrogen cycle. How much ammonia oxidation is from archaea, and how much is from bacteria, for example? No matter how big the part, archaea have important implications for the gardener who relies on the soil food web, or teams with it to provide nitrogen to plants. Decomposers. Archaea, like bacteria, are decomposers. They break down organic and inorganic materials, recycling elements needed for plant life. Some archaea get their nutrients by oxidizing sulfur compounds, which they can release from rocks. These archaea have been found where there is volcanic activity, such as Yellowstone National Park and Mount St. Helens in the United States, and in thermal areas of Iceland, Russia, and Japan. They are also abundant along seafloors, vents, where they either oxidize sulfur or use sulfur to oxidize carbon compounds. Either way, sulfur, a major plant nutrient, is freed and made available to other organisms in the web of life, and ultimately to plants. If you can tell me what is sulfur a primary, secondary, or trace nutrient? If you can tell me what sulfur is, I'll give you a giveaway. I have a plant for you. I have a sensitive plant, Mimosa paiduca, that I will give you. Not that one. This one back here. Okay. Methane is much more efficient at trapping heat in the atmosphere than is the more famous CO2. And we believe should be a greater cause of concern. It used to be that bacteria got the blame for methane. No longer. Then certain Archaea When certain archaea break down organic matter, they not only make nitrogen available to plants, they produce methane, a key component of greenhouse gas. One large source of archaea created atmospheric methane is rice production. Methanogenic archaea decompose matter in flooded rice fields, resulting in both plant usable nitrogen and methane. Given the scale of rice farming around the globe, this is a serious contribution. Methane levels have increased by about 150% since 1750, and it has been suggested that 10 to 25% of the methane emissions worldwide come from the activity of methogenic archaea, a tremendous significant amount. This group of archaea is not surprisingly under intense study by scientists trying to figure out ways to lower the amount of methane produced in agricultural activities. Holy cow, guys. Literally, we've been blaming cows and car emissions and cows for global warming, for greenhouse gases. It's actually extremophile fart. These Archaea bacteria in rice fields. That blows my mind. Most gardeners are now aware of the hypoxic or oxygen depleted dead zones that form where agricultural runoff of accumulates. Places like the waters at the mouth of the Mississippi River. Let's take a look at a rice field. Rice fields in southern China. And in the Gulf of Mexico. The nitrogen and phosphorus in this runoff allow algae and cyanobacteria to proliferate and thrive. They use up the oxygen, and what happily does not require oxygen and is standing by to decompose these organisms? Anaerobic archaea. Oh my goodness, Storm. Other benefits of archaea. Certain archaea are responsible for the production of archaeons. Archaeons. Archaeocins. Archaeonics. An extremely new class of antibiotics. Several have been identified and even manufactured, as the assumption is that there are hundreds more. The structure of these antibiotics is different from those produced by bacteria, so they act in ways existing antibiotics cannot. That there do not appear to be any pathogenic archaeons makes for interesting possibilities. That there do not appear to be any pathogenic archaeons makes for interesting possibilities. Perhaps they can be used against pathogens of both plants and humans that have become resistant or antibiotic have become resistant to antibiotics produced by bacteria. Archaeans are already an important source of specialized enzymes that work at high temperatures. One of those figures in the cloning of DNA. One of the features in the cloning of DNA. Others are useful for making organic compounds. Sewage treatment plants use archaea for purification, and the mineral industry puts them to use in some refining processes. Archaea enzymes are even included in some of the modern laundry detergent. Detergents. Hot water rinse, anyone? Haha, it's a gardening joke. Clearly, there will be applications using archaea for their byproducts for gardening. And there is always the possibility that archaea will provide nitrogen in artificial soil food webs established in such extreme environments as the Moon or Mars. Archaea, like bacteria and fungi, form relationships with other members of the soil food web. One such interaction is between the methanogenic archaea and protozoa in the digestive tracts of ruminants, termites, and other animals that consume cellulose. Ruminants? Ruminants? Ruminants. The protozoa break down the cellulose and release hydrogen, and methanogenic archaea convert the hydrogen to methane during their metabolic process. Even some protozoa are host to those types of archaea, and the human large intestine contains archaea that aid in digestion. Working mutually with bacteria that also live in the gut. Mutually. Mutually? Non-methanogenic archaea have been found in the soil of boreal forests, but we await further study to learn about their metabolic activities and other characteristics. As for the soil in your yard and garden, it is colonized by archaea, which inhabit the carbon-rich rhizosphere immediately around the roots of the plants. Since little else is known about the specific roles of these soil-based archaea, we will use this book and admittedly improper bacteria instead of the bacteria/archaea in future references. Let me read that once again. Since little else is known about the specific roles of those soil-based archaea, we will use this book, the admittedly improper bacteria, instead of bacteria/archaea in future references. We mean no disrespect to the archaea. Those present we dutifully acknowledge. Dutifully acknowledge. And dutifully, dutifully, acknowledge, and don't mean to suggest they are inferior organisms to bacteria, but without genetic identification abilities, we can't tell them apart. There is more to come. The diversities continue. Oh, the discoveries continue. Fully understanding the impact of archaea on the production of plant-usable nitrogen, the role they play in the creation of methane, and the scope of their interactions with other members of the soil food web will all help further the understanding of what goes on in the soil to help plants grow. Obviously, though, archaea are important members of the soil food web and can no longer be ignored. And that is the end of Chapter 4. We will be reading, this is going to be a fun one, Chapter 5, Fungi. Thank you all for coming on and hanging out. You've been watching Storm's Garden. And don't forget, it's our planet. So plant it.